Welcome to GeOMe demo number 1, where I'll show you how to upload metadata that describe massively parallel sequencing data that you will then be able to upload to NCBI. So we start off here on the home page, and it shows us in this workflow that the first thing we need to do is generate a template. Now this template can basically serve as a data management plan that you know ahead of time will be compatible with metadata collected by other labs. So we just go to the tools, go to generate template, and now you can see that it will actually just create a spreadsheet with a set of columns that describe your data. Now these columns have been selected from Darwin Core, which is a body of standards that is intended to facilitate the sharing of information about biological diversity. So you can be sure that your data, your metadata, will be compatible with data that are collected by other labs that are using GeOMe. Now we only have 3 required columns that are here in gray and automatically checked. We require something about the phylum, we require to know the principal investigator on the project, and we require a locally unique material sample ID, some sort of alphanumeric identifier of every single sample that is unique within your set of samples. There's a whole bunch of other recommended columns. These are columns that we would like you to provide information for, but we are not requiring it. So for example, I don't know anything about the sex of the surgeonfish that I'm about to upload, so I'll unclick that. There's a variety of other columns that I can use to describe my sample. So if we come down here to locality and habitat, we see a bunch of items that might describe from where we got the sample. And if I want to know the definition of any one of these columns, I can come over here to definition and click on it. So for habitat, we are using the environmental ontology standards, which is a classification of different biomes that a sample could have come from. And there's a controlled vocabulary that's very extensive and should cover most possible samples, most possible biological samples. So I select the columns that are important to me from locality and habitat. I select a few columns that are important for describing taxonomy. I select a few columns that are important for identifying the owner institution, the funding source, and the preservative that the genetic tissue is kept in. And I might have some pictures that I want to link to it, so I can keep that in the associated media category. When I'm all done with that, I come up here and I click Export Excel. And I can open that. And it'll first give me a set of instructions, so very clear instructions on how to fill out my metadata sheet. Then we come to the samples sheet. So this is where I'll actually be recording all of my information. Then there's a set of definitions for all of the fields that I selected, and all of the controlled vocabularies for all of the fields. So I can now take this spreadsheet offline, take it into the field, fill it out with the metadata that describe my sample data, and come back when I'm ready to upload the data. And now, cooking show style, we have some pre-prepared data from Dr. Michelle Gaither on Acanthurus olivaceus, which is a surgeonfish. And you can see we have data in all of our required columns. We also have data for all the other columns that we thought would be important for describing our data, our samples. And you'll notice that we do have some missing data in the coordinate uncertainty in meters. That's okay, that's just how life is, that's how research goes. We have that missing data. We'll close out of here and we'll go over to the validate and load data page. I can now browse to my metadata file, and it'll automatically pop up a map of all of the samples that have decimal latitudes and longitudes. And so now I can verify that the locations that the data say these samples came from are in fact where I think they came from. And I can click on each of these and see where each of the samples came from and verify that I do have my locations correct. I now push validate. Validation will check for duplicate material sample IDs. It'll check that the various columns meet the control vocabularies, and it'll check the required and recommended columns to make sure that they are present. In case a recommended column is missing, or if it's missing data, it will warn us, in this case, that coordinate uncertainty in meters has a missing cell value. I already know that's the case, but it gave me that warning, and now I might not have known it, and I could go back and correct it if I needed to. So now I can go to the Upload tab, and I'll be uploading a new set of data. So I want to upload metadata, and as well as FASTQ files that come from a massively parallel sequencing experiment. I would like to use a new expedition. Now, an expedition can basically mean any sort of data set. It can mean all of the data that come from a particular paper or project. It can mean all of the data that were collected by a particular person, or it can literally mean all of the data that came from a particular sampling expedition. So I want to type in a meaningful name for my expedition. So I will type Acanthurus olivaceus. That will be the name of my expedition. I will browse again to my metadata file. So now it is asking for a set of FASTQ file names. So I have a text file pre-prepared with a set of file names for the raw sequence data from each individual. So one file per individual, each file named after the material sample ID of that individual with .fq as the suffix. This is just a simple text file, so I'll browse to that. And then it asks for some information that is important to GenBank about, and the sequence read archive about our sequencing strategy. So I'll fill that out, single end. And there's definitions for all of these things if you're not sure which one your particular project falls into. In this case, we're dealing with DDRAD sequence data. So we're going to pick WGS, genomic, reduced representation. This was done on an Illumina HiSeq. And if I want to cite the particular protocol that I used, I can paste in reference to the DDRAD paper. And I'm all ready, so I can click upload. Oh, and it's asking me to verify that the samples are in the places where I think they should be. So everything looks correct there, so I'm going to verify that. Click upload. And it's giving me that same warning about the coordinate uncertainty in meters. So it's that same validation warning. It's something that I already know about, so that's okay. And it now provides me with a set of files, which I can download. And if I open these up, it will provide me with pre-formatted files that I can take directly to the sequence read archive, as well as a set of instructions that describe how to upload my data to the short read archive, as well as a link that will take me there. And that is how you upload metadata to GeOMe that describe massively parallel sequence data.